a gene has A's on both ends, and it could go in either in the forward orientation or the reverse. Okay, so it could go in both ways. Now, this is problematic if you're trying to make a fusion of your protein to, for example, the GFP gene. Okay, so if your insert, so if your gene that you're trying to clone in goes in the opposite orientation, and then you want to follow that on with the fluorescent protein gene, and it all has to be made as one protein, it's not going to work, okay? You're not going to make your protein because it's gone in the wrong way. So it's actually not that useful to use a single restriction enzyme when you're cloning, especially if you're making these types of fusion proteins that we talked about last week, because you want your gene or your DNA that you're inserting to go in a specific way. You want it to go in the forward orientation, so then when it gets transcribed and then finally translated into a protein, you're actually making your protein and then fused to, for example, the fluorescent protein or whatever tag that you're using. So this is what's called directional cloning, and this is essentially what we do most commonly, where we essentially use two different restriction enzymes. So one on one side of the gene at the start, whatever you want to call it, and one on the other side to actually insert into the plasmid. And that's when it becomes very important to have a multiple cloning site that you can choose two different restriction enzymes to use to linearize your plasmid. And what that means is that, for example, if you do, I think what have I used here? BAM and PST1. So, okay, they're not part of that thing, so there is no BAM in that one. Okay, so, oh, they are BAM and PST1. Anyway, so say you choose two different restriction enzymes. So say you choose this ZO1 and one of the other ones down here, BAMH1. Then you cut your plasmid. You still linearize it. It will still look linear, but you would have lost all the DNA sequence in between this ZO1 and this BAMH1. Okay, so it's not part of the, once you cut with that restriction enzyme and it cuts here, so you think your plasmid, circular one, and say that you choose another restriction enzyme as your second site, it means when you open up the plasmid, one side of your plasmid has ZO1 and the other side of your plasmid will be cut with BAMH1, and that small bit of DNA in between just gets lost when you run a DNA gel. So you end up with a plasmid rather than having one type of sticky end. It will have two different types of sticky ends because you've got two different restriction enzymes that you've cut your plasmid with. And you have to do the same thing when you're preparing your DNA that you're going to insert in your plasmid, so your gene or whatever it is that you're inserting. So, okay, so here we've got example of the gene at the 5' end, so at the start, before your actual gene sequence, you engineer, and we'll talk about how to do that later on in the lecture, you engineer a BAMH1 site. So that 6 nucleotide site that the BAMH1 restriction enzyme will cut will be located at the start of your gene. And then you engineer a different site, and say this is just PST1 as an example, is a different restriction enzyme site at the other side. So this is different to what we were talking about before, where we had two, like the same restriction enzyme on both sides of the gene. So we've got two different ones, and the plasmid is also restriction digested with the same two restriction enzymes, and what you end up with is a pair on one side of restriction enzymes and a pair on the other. So obviously once you cut your plasmid, it will have BAMH1 on one side and PST1 on the other, and same with your your gene that you're inserted. You have to also cut that with those two restriction enzymes, and you create that sticky end for BAM, so it will anneal to that sticky end of the BAM on the vector, and then you have the other sticky end of the PST1, which you have on your gene that you're inserting and also on the plasmid. So you have two different cohesive or sticky ends that then allow you to insert your your gene that you're trying to put into your vector. So this is, so what that means is that DNA fragment can only go in that way, because you can't switch it around, because the overhangs of PST1 are totally different to the overhangs of BAMH1. Okay, so these two totally different restriction sites, totally different sticky ends. So this could never go into, will never anneal to the PST1 site, and the opposite is the same. Did you have your hand up?
I was just going to ask, does that also remove the need for the dephosphorylation?
It does, but we still do it anyway, because it can happen still it does, but we still actually do that step in the lab anyway, because there are things that can happen in the tube with the ligase there that sometimes you can actually still get this strange thing happening. And that's another reason why we always DNA sequence the recombinant plasmids that we make, because we want to make sure that we've got the right sequence and nothing strange has happened in the tube or when we've transformed it into bacteria. So you would assume that removes the need, but we actually still do that step anyway as a normal step. Yeah. Yeah. Okay, so theoretically that shouldn't happen, obviously, but there are some that can still do strange things in very, very minor cases. Okay, so we've talked about this fact that I've been saying, you engineer a BAM site on the end of your gene and you engineer a PST1 site on the other end. How do you do that? So first of all, if the DNA that you're trying to clone doesn't contain, so you can use, if you're trying to clone a particular part of a gene, you might be able to find restriction endonuclease sites that border those sides of the bit of your gene that you're trying to make. So, for example, this is that TP63 DNA that we looked at from our Pratt class. And you could effectively, if you want to try and make trying to insert a particular part of that TP63 gene, you could effectively look for restriction enzyme sites away, outside of that area that you want to insert, and actually use those as the restriction enzyme sites. You would always have to have a look at your gene that you're trying to insert into a plasmid, and make sure that it doesn't have the same restriction enzyme sites as the ones you want to choose to do cloning. So that's going to be a problem, because it means that, say you want to choose the EcoR1 because this PCR 2.1 has this EcoR1 sites that you can choose for cloning. Okay, that's useful. Problem is that the TP63 gene has EcoR1 sites within it, which means that if you try and cut your TP63 gene to insert it into that plasmid, it means you cut up the gene. You wouldn't want to do that anyway. So the problem with doing it this way is that the restriction enzyme sites might be in inappropriate places, or you can't find the correct restriction enzyme sites that you could use. So before we talk about how you engineer sites, so how you create new sites at the ends of genes that wasn't there before in the actual gene sequence, I've just wanted to rehash again how you're actually doing your ligation in your practicals, because it's not restriction enzyme mediated ligation. So you're using this TA cloning method. Again, I think we've talked about it already, but you essentially make your gene with A's on the ends, because the DNA polymerase that you're using has what's called terminal transferase activity, and it puts A's at the 3' ends. And the vector that we're getting, we get it already linearized, and it's got these T's on the ends. And therefore, you just get a complementary AT and complementary AT. So this is also sticky ends. It's not a long sticky end like the restriction enzyme sticky ends. It's only a one base sticky end. It's not the same as a ligation, but one base sticky end ligation, I guess. And so it just allows your insert to go in much easier. The problem is that you have this issue with which way that your gene goes into this plasmid. It's not so important for this plasmid because it's a what we would call a intermediary plasmid. So you would end up taking your gene out of this plasmid after you've made a lot of it and then putting it into some other plasmid like this GFP one or something like that. So again, this is kind of removes the need to rely on restriction enzymes for cloning, and it just makes it a bit easier when we're doing the practicals. Okay, so well, I think my things are in the wrong sequence. So I'll come back. I was going to talk about how you make the different restriction enzymes on the ends of your PCR product, but I think they're right down the end, so we'll get back to that one. Okay, so what's very important when you're doing this directional cloning is because you want your gene of interest to go into your plasmid in a certain orientation, and that's why we do this directional cloning. So you want it to go in so it's red 5' to 3', okay, because your gene sequence, your ATG start codon, will be the starting methionine of your protein that you're going to make from that gene. And if you want to insert it into these types of vectors where you make a fusion protein, you essentially make a whole protein, so the same polypeptide chain, your gene of interest, and then fused to a protein, for example, the fluorescent protein. So it's very important to how to clone your gene in frame with the vector. So we're going to talk about how that actually works, or how you work out whether the gene that you're cloning in is in frame with the vector. So what this means is that when when your gene gets transcribed and translated in the bacterial cells, you want it to be read that your ATG is the start codon, and then the rest of your protein sequence for the gene that you've inserted, and then continue on exactly the same protein sequence for the fluorescent protein that you want to fuse it to. If you change the triplet codons, if you change that sequence, you're never going to make the right protein anymore. So it's really important to make sure that you're still in the same triplet coding sequence, so you're reading the triplet codons to make the protein that you want to make, and then fused to the say fluorescent protein that you want it to be fused to. Okay, so let's have a look how that works. So we're going to use so this vector has that multiple cloning site. You can see it's got a whole selection of restriction enzymes that you can use. So we want to insert our gene. We want to open up this plasmid at this Hindi3 site and at the BAMH1. So at one side, we will have a Hindi3, cohesive or sticky end, and at the other side of our plasmid and also our gene, we will have the BAMH1 sticky end. Okay, so let's have a look a little bit more, and importantly, once we've stuck our gene into this region between this Hindi3 and this BAMH1, once we cut our restriction enzyme, our plasmid with these two restriction enzymes, all this stuff in between goes away because it's not part of the plasmid anymore, because you've pretty much opened it up at two ends, and the stuff in the middle gets essentially gets purified away from the linear plasmid that you've made. Okay, so what's important is that if we insert our gene into this section of the vector, we have to make sure that it's reading in the same triplet codon to make the fluorescent protein as well. So how does that work? So when we make this green fluorescent protein fusion, we're making what's called a C-terminal fusion. So this is, once we make this as a protein, this is the N-terminus or the amino terminus of our protein. So this would be where our starting methionine is of our protein. So our protein is The gene of interest is in blue, is what we've stuck into the vector, and once we make that, we make the protein from that gene. So this would be the N-terminus of our protein, and then because this is a whole protein, this would then be the C-terminus of our full protein. Okay, we've made this fusion with a fluorescent protein. Okay, so it's what's called a C-terminal fusion protein. So the green fluorescent protein is pretty much tacked on to the end of the protein that we actually want to make. Okay, so you can see if you hone in on the actual sequence, the multiple cloning site sequence, you can see if you cut the plasmid with BAM H1 on one side, or Hindi3 on one side, so you can see if you cut it, it will be AAG, CTT. AAG codes for a lysine, CTT codes for a leucine. Okay, so that's the triplet codon that we have to read when we're actually making sure that where's my thing? Making sure that we have the correct reading frame. We have to continue this same triplet codon, or we won't get the right protein being made. Okay, so here is where our gene goes in to our vector. So this is our starting methionine of our TP63 gene. Okay, so this is the ATG start codon of TP63. So you've got a methionine there, and then this would be the rest of the DNA sequence, etc., whatever it is. Obviously, it's not the whole DNA sequence there. And then we would continue reading it with the same reading frame, so reading the same triplet codon as we continue. So then obviously that will read ATG, would be a methionine, that would be the start of our protein, and then every other triplet codon will be read in the same way. Okay, until we get to the end. So obviously our protein's not that short, it'll be much longer, but just as an example. So the last amino acid in our protein, so the last DNA triplet codon is TGC. So it codes for a cysteine. Okay, so that's fine. So that's the end of our protein, the gene that we've inserted into our plasmid. But now we want to continue to make the same polypeptide chain with the green fluorescent protein attached, and we want to be reading in the same frame to make the green fluorescent protein stuck to our protein. Okay, so how does that work? So we've got this, we're using BAM-H1 on the other side of our gene, and we cut the plasmid with BAM-H1, so you can see the coding, the restriction enzyme site is GGATCC. But you can see that it doesn't actually follow, so this in red is the restriction enzyme site of BAM. The problem with BAM is that it doesn't actually follow the triplet codons. So you can see the first G of BAM-H1 is part of this triplet codon, and then the last C is a part of this triplet codon. If we actually, this is part of the G, now will be part of making the GFP protein. So here we go. If we put on this GGATCC on the end of our gene, which we have, that will be part of that triplet codon. Then if we want to read in the same frame, then the GAT of that BAM will be the next triplet codon. The CC of the BAM will be the next triplet codon. But for us to read the start of the GFP gene in the same frame, we must add an extra DNA base. Okay, it can be whatever. In this case, we've added an A because we want to make sure we have the same triplet codon. If we don't have this A here, then this A of the ATG would be here, and then the next triplet codon would be TGG. That would no longer code for the start of the GFP gene, and you won't read it in the same frame. You can't make GFP anymore. So it's very important it's not just sticking your gene into a plasmid. You need to have a look at if you're making a fusion protein with some tag, and it can be at the start, the fusion, or at the end. You have to really look at these, whether what you're cloning in, your gene that you're cloning in, is actually in frame with the plasmid sequence. And so these are the things you have to look at. So it is does that seem clear? So if you don't add this extra A in here, so what it means is that you actually have to engineer this into your primers that you use, which we're going to talk about how you do that in the next slide. So if you don't keep the same triplet codon reading frame, you can't make the same protein. Okay, so if we don't, so we would be happy enough to make our protein here. So this is the end of our protein sequence. Okay, but if we don't, and we're using BAM-H1 to clone, if we don't actually add this extra A, then our GFP that we want to make stuck on to our protein